Okay, thanks everyone. Welcome to the webinar today. This is our monthly small systems webinar, Drinking Water Microbes 101. We have a very, very large attendance today and a lot of demand for this webinar. So before we get started, I do want to let you know that there may be some technical issues today, and we're going to do the best we can to push forward through those and make this a great webinar for you. So hang in there if you're experiencing sound issues. We'll try to let you know it's on our end, and we'll deal with those as they come along. So today, just so you know, the handouts, you do have a navigation bar, and in your navigation bar, you have a handouts drop down where you'll find a copy of the slides today. So for some reason you do drop off the webinar, chances are you won't be able to get back on, but you can call back in and follow along with the slides that were sent to you in an email and that are in the handouts right now. We will have a Q&A session at the end of both presentations today. You can type in questions for either presenters at any time during the webinar. So feel free to type those in and we'll get to as many questions as we can by the end of the webinar. Okay, and next slide, please. If you are, I know there may be a little delay in the slides as well. If you are looking for a certificate of attendance today and did not select that when you registered, go ahead and type in the questions box that you would like a certificate, or if you have anyone in the room with you today who would also like a certificate, you can put their name in the box as well. Next slide. Just a shout out real quickly to some of our upcoming webinars. Although they're not all water related, I think that some of these, maybe some of your colleagues would be interested in these. So we have a COVID-19 webinar coming up on the 27th, an air research webinar on air quality and healthy hearts, and a tools and resources webinar in June. So feel free to check any of these out. The links and registration are available on all of those pages. And next slide. And just one more piece of information. Some of you may be familiar with our annual drinking water workshop we hold in the Cincinnati area every year. This year we have decided to go virtual. So this is new for us. We're kind of excited about it. And we are working on the details of that right now. So registration is not open, but we hope to be announcing that soon. If you want details about the actual workshop and what it usually entails, you could click on the link here and get some information. Next slide. And with that, I'm going to turn it over to Tom Speth to introduce the first speaker.
Great. Thank you very much, Michelle. Can you hear me?
Yep, you sound great.
Oh, excellent. All right, so our first speaker today is Laura Boczek. Laura is a research microbiologist with EPA's Office of Research and Development, specifically the Center for Environmental Solutions and Emergency Response in Cincinnati, Ohio. Her research areas have focused on disinfection of various microorganisms in drinking water, as well as treatment of biosolids and wastewater. Recently, she's been involved with studies dealing with the fate and transport of antibiotic-resistant microorganisms, and she is also the chairperson of EPA's Pathogenic Equivalency Committee, which oversees novel processes that treat sewage sludge for land application. And with that, I'll hand over the electronic podium to Laura.
Thanks, Tom. So today I'm going to give you an introduction of sorts on microbiology for drinking water. It's going to be an overview of the different microbes that we've been concerned with drinking water throughout the years. So at the beginning, just want to let you know, microbes are some of the oldest life forms on Earth. They've been found about everywhere, from the depths of the ocean to the highest tops of any mountain. And they come in a variety of matrices, water, soil, and air. And there are 5 types of microbes that we recognize. Those are archaea, bacteria, fungi, viruses, and protozoa. This talk, I'm going to focus on the bacteria, viruses, and protozoa. Next slide. So when we talk about microbes, with just those three classes, this slide, I want you to see the diversity of these organisms that we're talking about. So each one of these classes are quite diverse from one another and have very different properties. So on this slide, this is a picture of a bacterial cell where you can see how simplified it is. There is no nucleus inside of the cell itself. It does have some things inside the cytoplasm, but everything is all connected together. The bottom picture there with that looks like a circle is actually a virus particle, and viruses are nothing more than just a protein capsid that has a nucleic acid, either DNA or RNA, inside of it. It actually cannot replicate on its own. It must infect a host in order to propagate and multiply in the environment. And then lastly here I have a eukaryotic cell, which is an animal cell. This is an example of something that a human has a cell similar to this. So we are more similar to our protozoans, who are also eukaryotic cells that I'll be talking about later on in this presentation. Next slide. So on this slide, I just want to give you a little bit of a comparison of what we're talking about size-wise with these organisms. These are very minute, teeny tiny organisms that we are talking about. So viruses are the smallest particle that I'm going to be discussing, followed by bacteria. Protozoa are the next one, bigger in size, and then finally helminths, which are worms that I'm going to discuss just a little bit. They don't really make the classification of microbes per se, but I did want to touch on them a little bit. And I just want to give you a comparison here with a human hair and a red blood cell for perspective. These things are much smaller than a human hair is. Next slide. So the very first part of my talk, I'm going to deal with bacteria. So bacteria represent a very diverse group of organisms. Most are non-pathogenic. However, there are some bacteria that are frank pathogens and others that are opportunistic pathogens. So what do I mean when I say frank pathogen versus opportunistic? Well, a frank pathogen is a bacteria that once it infects its host, it immediately causes disease, where an opportunistic pathogen may cause disease if the conditions are correct. So maybe if somebody is immunocompromised or has some other issues going on, then it can cause disease, but they don't always cause disease. And then also on this slide, microbiologists often classify organisms by their growth conditions. For instance, bacteria that grow only in the presence of oxygen are defined as aerobic bacteria, and then there's other bacteria that only grows in the absence of oxygen. We call these anaerobic bacteria. And then there's some organisms that can grow under both conditions, and those are facultative anaerobic bacteria. Next slide. So this slide just gives you an idea of how diverse bacteria can be. They can come in a variety of shapes and different sizes, morphologies. You can see on the right side of the screen all of the different morphologies that bacteria can be found in, from very small coccus to long spiral-shaped spirochetes. They also have different cell properties, which microbiologists have developed different techniques using stains and other growth conditions so that we can observe these organisms under a microscope and help to classify them even further. Next slide. And one of the techniques that a microbiologist uses is called a gram stain. And this is one of the very first tests that microbiologists typically conduct on unknown organisms in an environment. And basically what we do is we classify bacteria based on their outer lipid layer. So there are some bacteria that have a very thick peptidoglycan outer layer, and then there are some that don't. So we can call these either gram-positive or gram-negative. And based on this staining procedure, if the organism has this thick peptidoglycan layer, it will retain this purple stain that's used with, it's called crystal violet is the stain that we use. And if the cell is able to retain that stain and it can't be washed out during these later steps, then that organism is classified as a gram-positive bacteria. Gram-negative bacteria do not have as thick of a peptidoglycan layer, and because of that, then they do not retain that stain, and then they look pink on a slide. Next slide. So some bacteria also can have specialized structures. These structures give the bacteria some advantage for their survival in the environment. For example, some bacteria can form an endospore, which is a very resistant structure that allows it to survive in harsher environmental conditions than it normally would be able to survive. This is kind of like a dormant stage that some of these bacteria go into. And because of that, they're able to evade some of our typical disinfection processes that we use for water treatment. So when we think about water treatment, we also have to consider these kinds of properties that bacteria may have and make sure that our treatment is effective against them, along with all the other types of varieties of the bacteria come in. Next slide. So this slide, I just want to give you a little bit of insight to how bacteria replicate. So bacteria replicate very rapidly compared to other organisms, and because of that, they are able to mutate and change on a quicker basis than some other organisms that are of a higher classification could. And it's just this kind of response to the environment that can allow these bacteria to evade some of either treatments or antibiotics, other things that we use to try to eliminate them from the environment. Next slide. Microbiologists grow bacteria when we're using them in laboratories to study, in lab cultures typically, and we often refer to their growth in 4 different stages. And this is important for us because when we're conducting studies, where the bacteria is in its growth stage, a lot of times is very important when we're conducting tests, because we can get different results based on the life stage that the bacteria is in while we're conducting those tests. So the first stage is just the liquid media that's got all of the correct growth conditions for the bacteria to grow, and that's called the lag stage, where it's just getting ready. And then the second stage, where most of our bacteria that we're using to study is either in the log phase or the stationary phase. Log phase is when the bacteria are actively growing and doubling, and this is due to the great growth conditions. They're very happy in this media, and they're growing exponentially at this time. Then we reach the next stage, which is the stationary phase. This is where the culture is kind of in equilibrium. We don't really have much growth going on, yet we don't have death yet. And then finally is the death stage, where things start to die off because there's no good nutrients left. And then there's also acids and other things building up in the culture that actually start to kill the organisms that were once alive. Next slide. So this slide, I just want to give you just an overview of taxonomic classification for bacteria in particular. And and really all I really want you to take away from this slide is typically when microbiologists refer to the organism that they're working with or the organism that's a causative of some kind of disease, we usually refer to them as their genus and species name. And the genus name is always capitalized, and the species that follows it is not capitalized, and those are always italicized together. So for instance, you will hear a lot of people talk about Escherichia coli. So, or we shorten the Escherichia to E. So a lot of people also refer to the same organism as E. coli. It's the exact same organism. And another sort of confusing thing for people that aren't microbiologists is sometimes our species names are the same for different genus. So there can be some species overlap, but they would have a different genus name. So it's a different organism, so that's not to confuse you, but sometimes that does happen. And I like this little quote here from Lewis Carroll in Alice in Wonderland. You know, bacteria don't care what we call them, but you know, we give them a name for a reason to help us. So I like to put that up here with these kinds of slides. Next slide. So what I'm going to talk about next, I'm going to switch gears just a little bit and just specifically focus on these bacteria with respect to drinking water, and just go over on a high level their significance for drinking water. Next slide. So the first group of bacteria that I want to talk about are what we typically refer to as indicator bacteria. So these are the total coliforms, fecal coliforms, and E. coli. And coliforms in general are a group of gram-negative bacteria that all belong to the family Enterobacteriaceae. They've been used widely as indicators of treatment and disinfection. And why do we use these organisms? Well, we use them because they represent a class of bacteria that can be pathogenic and also are commonly found in waters that are polluted. So this makes them a good indicator. Now, there are some, with every good indicator, there's also some negatives to how well of an indicator they actually are. But by and large, these indicators have been used throughout the United States for our drinking water treatment as efficacy that our treatment processes are working. So there's a relationship between total coliforms, fecal coliforms, E. coli, and pathogenic E. coli that I don't know that everybody quite understands. So total coliform is like the big group of these organisms. And then you can tease that down further. So total coliforms are organisms that can ferment lactose at 35 degrees after being incubated for 48 hours. And then a subset or smaller set of total coliforms can also ferment lactose, but at an elevated temperature at 44.5 degrees. So not all total coliforms can survive that elevated temperature. The ones that can, we refer to them as fecal coliforms. And then an even smaller subset population of fecal coliforms are E. coli. And E. coli are actually a type of fecal coliform that also grows at 44.5, but typically has an enzyme called beta-glucuronidase, which microbiologists have developed specialized media to grow them on. We call these types of media selective media, and we're able to pull out E. coli from other fecal coliforms based on how they react in these media. So some of them fluoresce, some of them will produce a color change, those kinds of things. So when we use all of these different things, these different classes of organisms in different ways for our drinking water regulation. Next slide. So this slide, I want to talk a little bit about Legionella. I'm sure a lot of you are concerned about Legionella. I've heard a lot in the news lately about Legionella. So Legionella are bacteria that can hide inside of an amoeba, and they can persist in shielded from different disinfectants that we apply to kill them. And this graph here I have is the Legionella causes Legionnaire's disease, which is a pneumonia-like infection, and was first discovered in 1976 at a Legionnaire's conference. Next slide. So the next class of organisms that I'm going to talk about, bacteria-wise, are gram-positives. So everything that I've spoken about before this were all gram-negatives. This is a gram-positive bacteria. So enterococci are very small, and they are commonly found. There's some that are commonly found in the human intestine, and we use them as an indicator of fecal pollution. Many of these strains are antibiotic resistant and can survive in a variety of different temperatures and other types of constraints that our classical indicators cannot survive in. Next slide. So the next part of my talk, I'm going to switch gears a little bit and talk about viruses. So viruses are actually small particles that contain nucleic acids that infect humans, plants, and even some bacteria. Viruses cannot actually replicate on their own. They need a host in order to continue their life cycle. And most viruses are specific with respect to the host that they infect. We've all heard to nauseam in the news about the current virus around SARS-CoV-2, and that is a unique virus, and that it can't we know about with viruses. Next slide. So this slide, these pictures kind of look like alien life forms or something that we sent up to the moon. But actually, this is what a virus looks like if you were to look at it under a microscope. Viruses are in a very simple form, consist of a capsule that contains a nucleic acid. And once it's inside its host cell, it can hijack essentially the cell's machinery, the host cell's machinery, and then grow. It can replicate and then cause infection to other cells nearby it. Next slide. Okay. So viruses are classified as either enveloped or non-enveloped, much like bacteria, where we classify them as either gram-negative or gram-positive. An envelope virus contains a lipid bilayer around a capsid. This lipid is necessary for the virus to infect its host. If the bilayer is disrupted at all, it cannot replicate. Enveloped viruses, in general, because of this fact, are more susceptible to disinfecting practices that we use during typical drinking water treatment than non-enveloped viruses. The envelope also contains receptors on this protein caps. A lot of these bacteria are more efficient at being transmitted and causing disease than a non-enveloped virus. Non-enveloped viruses are sometimes referred to as naked viruses. And in addition to these different features, the viruses can either have a DNA or RNA, and the DNA can either be double-stranded or single-stranded. So there's differences, and we classify viruses based on if they're an RNA enveloped virus or if they're a double-stranded DNA, non-enveloped virus. Virologists use those terms to classify viruses. Next slide. Next slide. So this is, I'm not going to go into a lot of detail on this. This is just a, so I gave you the reproduction life cycle of a bacteria called binary fission. Viruses reproduce isogynous. Next slide. So when we talk about viruses and working with them in the laboratory, they are very different from bacterial cultures. And in fact, virus work is very difficult and expensive because these organisms have to have a host and they must grow on cell lines. So cell lines are sometimes difficult to maintain, and they have to have the specific cell line for a virus to infect it and for us to see how they behave in a laboratory. That a lot of these take that are necessary. It does make them more difficult to handle in a lab versus bacteria. A lot of scientists have looked into polymerase chain reaction, or PCR, to try to get around some of these culture problems that we see with viruses. The only issue that we have had with PCR is we're still not able to distinguish live versus dead with those types of tests. Next slide. So just like our viral or bacterial cultures, we also look to use indicators for our viral organisms, and we use them because they're more abundant and they're much easier to grow than some of the viruses that we are concerned with. And we use them as a way to track disinfection and other types of treatment to see if we're able to remove these things. Next slide. So for this next slide, all I want you to take away with is that there are the most common bacteriophages that you will come across with drinking water treatment are somatic colophage and male-specific colophage. They do look a little bit different on a plate. So those plates that I have on the top there are somatic colophage versus a male-specific colophage. One makes a bigger plaque. Those little circles are called plaques where the E. coli was not able to grow because the virus infected it there. Next slide. And some of the advantages that we have to using colophage as indicators are the same as using indicators for bacteria. They're easier to use, and they typically are found in higher numbers than our viral yet again. And now we're going to focus on how they relate to drinking water. So protozoa are actually eukaryotic cells, and they can either be free living, meaning they don't need to live within a host, or they can be parasitic, meaning that they need a host to continue their life cycle. You'll see in some of my later slides that some of them have very complex life cycles where they actually go from the host cell to an environmental strain matrix, and then back to a different host to infect. Protozoa in the laboratory, to mimic that in the laboratory can be challenging at times. Next slide. I'm going to continue on. So can you go to the, okay. So this slide I want to give you here is, it just gives you an idea of the different morphologies and different types of protozoa that are out there. These are actually unicellular eukaryotic organisms. So these are more related to a human cell than what our bacteria cultures are. And like I said, they can be free living or parasitic. And this bottom Venn diagram just gives you some of the commonalities that we have between prokaryotes and eukaryotic cells. Next, so there are multiple hosts and have, to the next slide, they can have different forms and stages in their life cycle. So on the bottom slide here is an amoeba. So for instance, an amoeba can form a cyst, which is a very hard resistant form of the amoeba, and it can help it survive in environments where there are harsh conditions that would normally kill it. And then when those conditions subside, that organism then can transform back into its amoeboid trophozoite phase. And also can go into a flagella-like form where it can swim and move more efficiently than it could in its trophozoite stage. Next, of these organizoa can form cysts, which are like the bacterial endospores, are a more dormant, hard resistant-like stage that help them to resist treatments and disinfectants. On the next slide, I have lots of different pictures of Giardia cysts versus a Giardia trophozoite, so you can see the difference there. Cryptosporidium cysts, another organism that we're concerned with in drinking water, Microsporidia, which also can form these cysts. Next slide. Then the next slide, I have a pictogram of the life cycle of Giardia and Cryptosporidium. And what you can see from this slide is the very different and unique properties that these organisms have. So they can form a cyst stage where they can either be infective, and then they're in a different stage when they're diagnostic, and they both, it's just an example of how complex these organisms really are in causing disease. Next slide. And lastly, I want to talk about parasites in our country than what they are today. A lot of these are not as common here. They're more common in other countries. But they also have very complex life cycles and some in their life. And our treatment has to take care of all of those stages. And that is the end of my talk.
Great. Thank you very much, Laura. Can you hear me?
Yep.
Excellent. We're getting better at this. So, thank you, Laura. Also a note to the people listening in, if you have any questions for Laura or Jennifer, who is our next speaker, while Jennifer's talking, please put them in the question box right away. That helps us organize the questions for after Jennifer's talk, when we'll do questions for both speakers. So please, if you have any more questions for Laura, or as you hear, Jennifer's her best. Jennifer is a microbiologist with EPA Water and Drinking Water Support Center in Cincinnati specifically. She supports many microbial aspects of the federal drinking water program, including technical support on a number of programs, such as the alternate test procedure program. And she also serves as the Part 9000 coordinator for standard methods for the examination of water and wastewater. She's been with the Jennifer since 2001. And with that, I'll give the electronic podium over to Jennifer.
Thanks, Tom. Can you hear me?
Yes, I can. Excellent. Thank you.
Okay, great. Okay. I've had to move to my landline because my mobile phone is acting a little bit funky. Okay, so I'm going to talk today a bit about how we came to regulate the microorganisms that in drinking water that Laura just talked about, and how we've used regulation to help make our water safe for everyone drinking it. So as hard as it is to believe, given all the information that Laura just presented, people prior to the 20th century, they didn't understand how disease was transmitted or how it occurred. They believed in part it was bad luck, and in part of it was bad air. When you see pictures from people, from doctors in the plague, they'll have those bird masks on, and that was in part so as not to breathe the air. So they had an idea that it was contagious, but they didn't quite understand how. In 1676, Anton van Leeuwenhoek, he developed the first microscope, and I have a picture down at the bottom corner here. And he was the first person to actually see something in the water when he looked at a drop of microbes. And he tracked all the cases of people who were drinking water from this one pump on Broad Street. So he went down and he took the handle off of that pump. And when he did that, the number of cholera declined. And so that was one of the first bits of information that disease could be transmitted through drinking water. With that information, the people started to gather more bits of information about the idea that this might be caused by not just bad luck. Disease might actually be caused by microorganisms. And around the same time as Dr. John Snow took the pump handle off, we had Louis Pasteur, who was working on the germ theory of disease. This was proven by Robert Koch in the late 1800s. But despite all this evidence, people still didn't accept the fact that there were germs in the water. There were bacteria and other microorganisms that were responsible for the disease. Next slide, please. Can I have the next slide? Next slide. You hear me? Around that same time, there was the Industrial Revolution in cities. So what happened is people were moving out of their farms and into the cities, and they needed water to drink. So community water systems began to be developed. So you had people beginning to realize that the disease wasn't just bad luck or bad air, and you also had people moving closer together, which being they are more susceptible to disease, but they also were all using common supplies. So you had people starting to recognize with John Snow's understanding that that disease could be spread through water, that people needed to maybe keep their wastewater and their water safe. They also had more waste, so people developed the toilet, which then became hooked into sanitary systems. So around this time, there were a lot of typhoid fever outbreaks, and people began to have big problems. About the number of people that die today in car accidents were about the number of people that died of typhoid fever. So in the 1800s, this all became, their drinking water wasn't that actually started to chlorinate their drinking water supplies. Next slide, please. Can you hear me? Okay, thanks. This is just a graph showing that the number of typhoid cases throughout the year. So you can see that in 1900, there were about 30 deaths per 100,000 people. And it was about 1910 when drinking water treatment began, and the number of deaths of typhoid fever begins to dramatically decline. But if you notice, what's important on this graph is that antibiotics didn't come around until it weren't commercially available until about the mid-1940s, after World War II. And so drinking water treatment was named one of the greatest achievements of the 20th century for that reason. It's the water treatment in this time frame. To the next slide. Can you hear me?
That's terrible.
Sorry. Okay, so people realized all this, but at the same time, there wasn't really a strong federal regulatory program. Obviously, as people were just beginning to realize the problems that can occur if your drinking water is contaminated or wastewater can get into your drinking water. And this, you know, our country was still relatively new at that time and still hadn't figured out all the things that it could regulate. And recall, at that time there was a lot of sentiment of people didn't want big government, and they didn't want, the states wanted a lot of autonomy. So there wasn't a lot of federal government oversight at this point in time. The first step of the federal government into any kind of disease control was in 1878 with the National Quarantine Act. And that was where the government, at the time it was the Marine Hospital Service, they came in and said, you know, we don't want people that are sick getting off of these vessels that are coming into ports. And that was the first time that the federal government kind of stepped into what had previously been, okay, the states have their individual rights, but we're going to override and control infectious disease on things that cross state lines. This applied only to things that provided water across state lines, so like boats or trains. And under this rule, they banned what was called the common cup, which, I mean, I'm a microbiologist, so admittedly I'm a germaphobe, but I can't imagine the idea of the common cup where there's just a cup and everybody drinks out of it to get water. And that was the regulation that they used to be able to ban that. And finally, in 1914, the United States Public Health Service developed the Interstate Carrier Program. And this is important because it's the first time that they set limits on the number of bacteria that could be in water. Prior to that, there really wasn't any kind of standard. This was the first standard that we had, and it applied only to interstate carriers again, but they set the limit of 100 organisms for a bacterial plate count. And this was later adopted by many states, since it was kind of the first standard that was set. States used it because they didn't know individually what the limit should be. Next slide, please. Next slide.
Hi, Jennifer. This is Michelle.
Yes.
Can you hear me? I
Yes.
just want to let you know that there's just a little bit of a delay when your slide advances. So it'll just take a second to catch up with you.
Okay. I'm not sure if you can hear me though. No audio issues.
Yes, perfectly. Yeah.
Okay, good. Okay, good. Okay, so in 1942, the United States Public Health Service required regular microbial sampling, and this was based on the number of samples per month, or based on the community require this. It was the best that they had, and it's easier for everyone to kind of use the same standard, and it was a good standard for everyone to use, and it became consistent. In 1969, the Public Health Service did what was called the Community Water Supply Survey, and what they did is they went around to a lot of communities throughout the country, and they examined the water for coliform limits and for, in some cases, HPC. And they studied what the water looked like and how frequently it was being monitored. This was done right before the creation of the EPA, and those data were used to formulate some of EPA's early understanding of how contaminated our drinking water was on a national basis. So, at the basin of like the Mississippi, which collects water from the Ohio and other things. So, this was the impetus for the EPA to take over the drinking water regulation, which had previously been done by the Public Health Service. Next slide, please. So what can happen if your drinking water is contaminated? You can have two main types of outcomes. The first one could be a short-term exposure. Generally, microbes in your water will cause something like diarrhea or nausea or other short-term effects. You will get over them. They may be deadly, but generally you get sick more immediately, and it's something that will subside, versus chronic health risks, which tackle through the different types of drinking water regulations that we have. Next slide, please. So before I go into the regulation, I just want to explain quickly a little bit about regulations for those of you that don't know. A statute is enacted by Congress and signed by the President. Regulations are then written by an agency with the authority coming from the statute. So for EPA would write regulations based under a statute that Congress has given them. The regulations are published in the Code of Federal Regulations. And finally, because the agency can't always put absolutely everything necessary in a regulation, because the regulation might be 1,000,000 pages long in some cases, the agency writes policies or guides as to how the different types of documents that the agency puts out to help protect drinking water. Next slide, please. So there is one, well, more than one, but one in terms of drinking water, one landmark legislation that's called the Safe Drinking Water Act, and it was enacted in 1974. And the purpose is to protect drinking water supplies throughout the nation. And what this act did is it authorized EPA to set federal enforceable health standards for contaminants in drinking water and public water systems. So what's important there is that EPA does only have the authority to regulate public water systems, and CIDWA gives a very specific definition of what a public water system is. It's defined as serving at least 25 people or with 15 service connections. Hold on a second. On 6, is it to meet those criteria, a term called primacy. And what primacy means is that the state will accept responsibility for enforcing the regulations on behalf of the agency. And primacy really works well because it allows the state flexibility to be more stringent than they need to be with our federal regulations. So I view federal regulations as a one-size-fits-all, but sometimes the one size doesn't really fit all, and the states want to make it more stringent, and they can do that under the terms of primacy. They just cannot be less stringent. The Safe Drinking Water Act of 1974 also told EPA to start writing national primary drinking water regulations, and those would give more specific requirements around the contaminants that could be in water and at what level. What's important to note is that CIDWA has been amended many times throughout the years, and I'm not going to talk about all these amendments. I'm just going to talk about a couple that impact what you're doing today and what we're doing today the most. Next slide, please. Okay, so what does EPA drink water regulations? A variety of microorganisms, which Laura talked about, crypto, giardia, heterotrophic plate counts, total coliform, fecal coliforms, which are sometimes referred to as thermotolerant coliforms, E. coli, turbidity, and viruses. Viruses in general, not specific viruses. I'll talk more about that in just a second. Disinfection box, that'll give you more details about, like, for instance, all 54 of the organic chemicals that are regulated. Next slide, please. Okay, the first amendments, one of the Safe Drinking Water Amendments was 1986. The reason for this amendment is because EPA was a little bit slow on the uptake of getting the national primary drinking water regulations written, and Congress said, EPA, you need to work a little faster, so we're going to amend this, and we're going to give you some specific things that we want you to regulate. The amendments, under these amendments, it describes, it tells EPA to MCLs and MCLGs, or treatment techniques for specific contaminants. MCLs are the level that can be in water, that's enforceable. MCLGs are the goals of levels of chemicals that you would like to have in water. The goal, you would like to get to that goal, but sometimes it's not feasible either by treatment or by the goals are not enforceable, but MCLs are. Treatment techniques are where we tell people that the regulation will say, for instance, you should apply this treatment, and in doing so, that will knock out like this many cells or this type of contaminant. So you don't actually have to monitor to show that it's there. You just follow that treatment, and then the assumption is everything. There might be things that are not easy to measure or to make them. The amendments also required EPA to regulate 83 specific contaminants, including total coliforms and the heterotrophic plate count. Next slide, please.
Hi, Jennifer, this is Michelle. Sorry to interrupt you. I'm just going to jump in for 1 second before you continue. Because we've actually reached the top of the hour, so things started to It's okay, Jennifer. I just wanted to make sure everyone knew because some people would have to leave. Okay, go ahead and continue.
Okay. Thanks, Michelle. Okay, I think the information on this slide I touched on the last slide, so we can go ahead to the next slide. Okay, so a little bit before I start talking in depth about the regulations, I just want to talk about what each regulation has in it, because this is really important. I think for people that don't work in the regulatory field, they seem daunting. They did to me when I first started. I thought, holy moly, how can anybody read this whole thing and digest it? I think when you learn a little bit about what each rule tells you, it makes it a little bit easier to understand and digest it and get through it in a way that you can understand what it applies to you. So the regulations have to tell you what technique you're going to use to treat what's acceptable, how to take your samples and how long you have to get them to the lab and what temperature, if there is one, they must be held at. It's important for microbes because they can change when they're being shipped to the lab, like the number, they could die off or they could increase if you don't refrigerate them. Some rules don't require refrigeration, but some do. And finally, the rule has to give you the method to use to measure that contaminant. We can't just have people using whatever method they know, because then the data that we gather would not be consistent, nor would we be able to provide assurance to all the consumers of the water that the water had been analyzed in a consistent manner with, say, the next town over. So all these things are in each regulation. When you go to the regulation, you'll see it kind of broken down into chunks. And these are the core things that are in it. Now, you're going to find other things in each regulation, like economic analysis, and all these and health effects data. You'll see all these other things, but at its core, these are the kinds of things each regulation has to tell you. What do national primary drinking water regulations cover? They cover finished drinking water, but what confuses some people is that in some cases they cover source water that will become drinking water. So that water is not drinking water yet, but it makes sense that you sometimes regulate that because you want to make sure what kind of treatment you may need or how contaminated your source water is so that it can become the best drinking water possible. Because once if you have problems in your finished water, that's harder to deal with than problems in your source water, in some cases, not all cases. And finally, as I mentioned before, CIDWA gives the individual states the opportunity to set and enforce their own standards if they're at least as stringent as EPA's national standards. They cannot be less stringent, but they can certainly be more. Next slide, please. I usually work on the analytical method, so this is definitely my comfort zone. But for each regulation, as I mentioned, the regulation has to give you at least one method to look for that bacterium or that parasite or whatever. The method will also describe some QA, QC. Now, you might be sitting there going, but I know there's 15 methods approved for coliforms or whatever there are. And that's right, there are. The rule has to give you at least one method. And in rules such as the total coliform rule, which has been around for many years, I think we have 15 coliform methods now. And that's because throughout time, vendors have developed new, fancier, faster, better methods. And the agency has mechanisms by which we can approve those methods once they're developed. So we can't regulate something without a detection method for it. But once we have at least one method, if there are more available, we'll approve those and give laboratories and water systems more options for how they might want to have their analysis performed. In doing this, you can rest assured that we determine through our processes that the additional methods we approved are considered equally effective. They're pretty equivalent with one another. We don't want to get in a situation where we have methods that are straying wildly from one another, because then that calls into question some of the data that we would generate. And finally, a point that I always harp on is your laboratory has to use the approved versions of the method. So in the regulation, it will give the name of the medium or the method, and it will then say, like, for instance, some kind of tracking, like a date or a version, and that is the version that must be used by certified laboratories. The other thing the CFR tells us, which I didn't highlight earlier but is important to know, is that any samples taken for compliance with national primary drinking water regulations, they must be analyzed by a certified laboratory. And by a certified laboratory, I mean one that has been certified by the state that it's doing business in, or accepted by the state that it's doing business in. Next slide, please. Okay, in some more significant amendments made to CIDWA, and in 1996, what happened is Congress said, in a nutshell, that EPA wasn't being transparent enough in how they were deciding what to regulate and making their regulations. So, under the 96 amendments, EPA developed a new framework for how they would rate, and next is a UCMR, which some of you may be familiar with. The next step is in the 6-year review. These are all designed to have priorities based on adverse health effects, occurrence, and estimated reduction of risk. So at the CCL, we have like a pool of things we think could be contaminating drinking water, could be of concern in drinking water, and then some of those may be monitored for under the unregulated contaminant monitoring rule. Once we have data on those, we then determine through a process called regulatory determination if it's warranted to develop a regulation for those items. And finally, if we do regulate those items, all of our regulations are re-examined every 6 years to determine if they are still effective the way they're written. One of the tasks given to EPA in the 1996 amendments are to develop rules that balance the risk between pathogens and disinfection byproducts. Of course, it's easy to kill all the pathogens if you just pour tons of disinfectant in, but then everyone gets sick from DVPs. So that's one of the things, as I talk about some of the newer regulations, you'll see that there's a balance there. The 96 amendments also called for increased public awareness, where you'll get consumer confidence reports, sometimes in the mail, sometimes on your water utilities website, and also databases that have data about contaminants in water. Next slide, please. Okay, this is just a schematic that shows you how these processes work. The way I described them is kind of in a vacuum, but they're not. They feed back into each other with the UCMR data feeding back into CCL and such. Next slide, please. Okay, the first set of regulations which are important. The way I talk about regulations and the way it makes sense in my head is to first talk about water before it enters the treatment plant, and then so raw or surface or source water, and then I'll talk next about the regulations that cover treated or finished water. So the first set of rules are called the surface water treatment rules. It's a suite of regulations, although there is actually a rule called the surface water treatment rule, which I'll talk about next. But these are actually a suite of regulations designed to provide protection from pathogens in surface water. And surface waters are like rivers, lakes, streams, and they are at risk for high amounts of microbial contamination because they get treatment, they get runoff, or in the case of like Cincinnati where I live, sometimes the wastewater plant can't handle the demand, so sometimes raw wastewater is dumped into the Ohio River. Stormwater runoff, agriculture feedlots, and just wildlife. There's badgers and there's birds, and they're all pooping in that water. Under these rules, you'll see, as I just mentioned, that balance just adds so much disinfectant that you're just killing everything, but then people also are getting sick. So you want to balance out the minimal risk with the disinfection byproducts. And finally, these rules were the first that required water systems to filter and disinfect. Some water systems don't filter, though. There are criteria. I'll talk about that in just a second. that they can't avoid the filtration part if they meet the criteria, but everyone who uses surface water has to disinfect. Next slide, please. Also, I put a website there in case you want more information about how these rules all work together and how they all work together to protect surface water. So this is a list of the rules that are under this umbrella. I'm only going to talk about the original surface water treatment rule, which was promulgated in 1989, and I'm going to talk about LT2, which was promulgated in 2006. But the other rules are the interim enhanced surface water treatment rule, the filter backwash recycling rule, and the long-term one enhanced surface water treatment rule. Long-term one serves to set limits around crypto, and LT2 builds on that, and the interim enhanced, I'll talk about those in just a second. Next slide, please. Okay, so the original rule was called the surface water treatment rule, and it is still very much in effect today. And it applies to all public water systems using surface water, or what's called GUDI, which is groundwater under the direct water, which if it's groundwater under the direct influence, it's considered to be surface water for the purpose of this rule. This requires the use of sufficient water treatment technique, either removal or inactivation, to reduce Giardia by 99.9% and enteric viruses by 99.99%, which is a 4-log removal. This rule, however, in case you're sitting there scratching your head going, but I don't take samples at my water treatment facility for Giardia or enteric viruses, you don't, because as I mentioned, one of the things that can be applied under our rules is just what's called a treatment technique, which means you don't monitor, you just follow the recommended treatment. And that's what this rule is. The water systems don't monitor for viruses or Giardia. The rule gives them recommended treatment techniques, and if they're following that, then they are achieving these goals of removal of these pathogens. The disinfection is what's required here. As I mentioned on the last slide, it does require water systems to filter their water unless filtration avoidance criteria are met. Filtration avoidance criteria are designed to show that the water is pretty pristine, and they then can just disinfect. There are very specific criteria, and I believe some states do not even allow the filtration avoidance criteria. Some states just said everybody's going to filter. But at a federal level, those are present in the rule. And I've got a website here listed in case you want to go read about those. Next slide, please. Okay, this is the filtration by avoidance criteria. It's just showing you have to do a lot of monitoring. You have to disinfect. You have to have chlorine residual at the entry point. You can't have had any outbreaks. You have to have no more than 5% at 0.5 NT turbidity, and you have to monitor weekly for total coliforms and fecal coliforms. Okay, next slide. Okay, LT2 is the next rule I'm going to talk about. This is sometimes referred to as the crypto rule. This rule is in that suite, as I mentioned, and this is a rule designed to provide high-risk systems additional treatment for cryptosporidium. So it builds upon, as I mentioned, the requirements from the interim enhanced and LT1 rules. And in bringing this rule in, it will also increase protection against other pathogens, because in this rule, water systems are placed into bins based on their risk for contamination from cryptosporidium, and they will provide additional treatment to mitigate the risk from crypto. And in doing that and providing that additional treatment, they're also providing protection from other organisms. This rule also does contain provisions to mitigate the risk from uncovered finished water reservoirs. Under this rule, water systems will monitor for cryptosporidium. This rule had what's been termed like a staggered implementation. In doing that staggered implementation, it meant that systems were broken down by sizes, and depending on how many customers they served, that's when they monitored and what they monitored for. The large systems under this rule monitored for crypto and E. coli and turbidity, and those E. coli data were used to set what were termed the trigger values for the small systems. So next slide, please.
Hey, Jennifer, are you having trouble? Are the slides not advancing for you?
It just advanced. It's taking like a long time, so maybe, I'm not sure if it's taking a long time for everyone else or just for me. It's taking like a minute, I feel like, for them to advance.
Yeah. Tom, are you
okay.
You got it?
Yeah, perhaps Jennifer, since it'd be great to have some time for questions, if you could wrap up in the next few minutes, I think then we could get to a few questions. Thanks.
Okay. LT2 began in 2015 and it will be wrapped up next year. And in 2017, the agency did come out with this determination that in the 6-year review 3, that this rule will not be revised. So and this is due to the fact that round 1 levels were lower than expected. Next slide, please. Okay, I'll just talk a second about the TCR. This rule was promulgated in 2013. That revised the 1989 rule. This rule affects every water system in America, so it's one of the few that covers non-transient, non-community water systems as well as transient, non-community water systems. It establishes an MCL for E. coli. It uses total coliforms in E. coli to initiate a find and fix approach for potential fecal contamination of a water system. I've given a web address there for you to go and look up more information about this rule if you wish. Next slide, please. Okay, and this rule uses the number of people on a water system served to determine how many samples they may take. And this rule also doesn't require, but the data is best if you take it throughout the distribution system, so you understand any issues you may have throughout your system. Next slide, please. Okay. One of the biggest changes from this revised rule from the old rule, there is no longer an MCL for total coliforms. Instead, there is an assessment that's done if the system reaches the number of, or exceeds the number of total coliform positive samples they can have. They do an assessment. This rule does state, as you take your samples, any sample that is positive for total coliforms is further tested for E. coli. And if you get a positive, this system sends you into additional samples that are taken. So the way I think of this rule is that if you get a total coliform positive, you're either going to take a lot of samples until you get triggered into assessment, or the problem you had is going to go away and you're going to be back to getting negatives. Next slide, please. Okay, this is just showing you, this is how you get an MCL violation. This is a nice pictorial I found. Next slide, please. Okay, the next rule I'm going to talk about is the groundwater rule. This is the last rule I'm going to talk about. It's promulgated in 2006. And this is kind of the counterpart to the surface water treatment rule. This is for water systems that use groundwater as a source for their drinking water. And this rule is really a risk targeting approach. It requires states and water systems to do like assessments of their well to determine how risk they are for contamination. This rule is nice because it also dovetails with the TCR. So if you start to have TCR problems, you can also have to go look at your source water to figure out if that's where your distribution system problems are coming from. This, again, only does apply to water systems, not wells, because private wells are not they're outside the authority of the agency. Next slide, please. There's two kinds of monitoring, triggered monitoring and assessment monitoring. I just mentioned if you get total coliform problems, you're triggered into monitoring. Assessment monitoring is not really a requirement of this rule, but it does require the states can require assessment monitoring if they have concerns about the risk of the groundwater system. Next slide, please. And this rule is different. What makes this rule different is on this slide right here. This rule does not use total coliforms as indicator organisms. Instead, it allows the use of what are termed fecal indicators. And the water system, or in some cases the state, chooses for the water system, they can use one of these three options: E. coli, enterococci, or coliphage. What makes this interesting to me is that the agency is the first time that have used coliphage. What's unique and what's nice about coliphage is it could provide kind of a model for how enteric viruses would persist and be mobile throughout a groundwater system. So based on the information I have, I'm not sure that I think a lot of states or water systems are using the coliphage option, but I definitely think that's something that the agency is collecting information and interested in seeing how that may work out as an indicator instead of our traditional totals and E. coli construct. Next slide, please. Okay, so this just shows you where where you can get all the information if you're interested in learning about the methods approved under these rules. This gives you Code of Federal Regulations citations. It'll tell you the methods, but you also needed to go to Appendix A to Subpart C, as that has additional approved methods there. So if you have any questions about that, you can email me. And I've given my contact information on the next slide. If you have any questions, I'll certainly answer what we can now. I apologize for any issues that we've had that have slowed this down, but you can certainly email if you have any questions about these.
Okay, great. Thanks, Jennifer. Can you hear me?
Yes, I can hear you.
Oh, excellent. Well, thank you for an excellent talk and with trying circumstances. Every time we do this of late with these broadband issues, we learn something new, and we're going to try to clean it up for next time a little bit. So I thank our audience for hanging with us. A lot of people still have, I've noticed. So thank you so much. We're going to get right to questions. Laura, give up.
So the main thing that we use that distinction for is it gives us, so it kind of tells you what kind of infections you may be dealing with. If we're talking about gram-positive versus gram-negative, gram-positives have thought to be a little bit more hardy than gram-negative. However, gram-negative bacteria can also cause some pretty nasty diseases. So it's just one of the tools that we use to try to identify an organism and then find proper treatment for.
Okay, great. Here's another one for you, Laura. On slide 17, you discussed total coliform and fecal coliform, E. coli. Can you describe which is a subset of what and what temperatures they proliferate at?
Sure. So total coliforms is the big overarching group of these organisms. So all E. coli are also total coliforms, but not all total coliforms are E. coli. So the big circle is the big picture group of the organisms, and total coliforms very simply are just organisms that are able to grow at 35 degrees and ferment lactose within 48 hours. And then we can make that a smaller set by seeing which of those organisms can grow at 44.5. And those organisms that can grow at 44.5 and ferment lactose are considered fecal coliforms. They would also be total coliforms, but there's not going to be as many fecal coliforms as there are total coliforms. And then a smaller subset of the fecal coliforms is a group called E. coli. And E. coli are those organisms that grow at the elevated 44.5 degree temperature and also have an enzyme called beta-glucuronidase, which we use with our microbiological media.
Okay, great. Thanks. Here's another one, then we'll go to Jennifer. So Laura, on slide 24, just to clear something up, on slide 24, it says DNA, RNA methods can't detect for the presence of live infectious virus. Do you mean that they can't detect live viruses, or they can't distinguish between live and dead?
So they our PCR-based protocols do not determine if the organism is live or dead. All it can tell you is if the organism's nucleic acid signature. And when we do these PCR tests, they're really only looking for a very small section of the DNA. It's not going to look for the entire DNA signature line to see if it's all there and intact. It only looks for a very small portion of that, and it can tell us basically if that organism was there. We cannot determine if the organism is infective or if it can continue to cause disease.
Okay, great. Thanks, Laura. And a few quick questions for Jennifer. So Jennifer, what is assessment monitoring? What could trigger it? And why would somebody want to do it?
That is not a requirement of the rule, as I said. That is something that the states can implement. But I think if the triggered monitoring may not help you, a state may require that a water system go to assessment monitoring, which is more of them trying to understand what their risks are and how their wellhead may be susceptible to contamination from something, for instance, as septic systems, or maybe if there's runoff from an animal feeding station or some wildlife. Wildlife probably isn't typically going to be enough to contaminate, but so it's just a way to obtain more data to determine how to make sure that there is not any more contamination of this groundwater source. And that's just a tool that's in the rule for states to use if they need to provide better quality of water to the consumers.
Okay, great. Here's one. Because camouflage is more difficult and more expensive, what is the benefit for a public water system to monitor using that fecal indicator?
Camouflage is definitely more of a challenge because the other fecal indicator, well, specifically E. coli, there are some pretty cheap methods available, and the states are already geared up to and the labs are already geared up to look for E. coli. So I agree, the camouflage methods are quite a departure from some of the more simple methods used for total coliforms and E. coli. But the benefit, in my eyes as a microbiologist, is that if there's any concern about enteric virus contamination, the coliphage are about the same size, and they're gonna be able to be mobile throughout the wellhead. So if you have one like made out of karst or what have you, you want to understand maybe how viruses could travel through the water system. You'd want to look for an indicator that could travel the same way as well. And bacteria and viruses, as Laura described. And as we know, the correlation between the outbreak data that we have does not show that 100% there's always a correlation between bacteria and viral outbreaks. So this is an option for people who have concerns about that to understand how viruses may work, may cause problems throughout their wellhead or around their water system.